Good morning, everyone, and welcome to this third class. And I find it worthwhile to mention that we are following the guideline with the syllabus given in NEP UGCF, and we are tracking the paper of genetics and molecular biology for BSC Life Sciences first year. So this becomes the third lecture on genetics and molecular biology, and today what I plan and what we'll be studying today is the induced mutations. And while teaching, my focus would be on how and why mutations are induced by chemicals and radiations, because mutations are random. Very often they may not produce the change which we want in the cells. So we'll be mainly focusing on induced mutations, but our meaning, if you straightforward join for this third lecture, it would be hard for you to follow, because for this one you need to come back viewing the previous two lectures on mutations. In the first lecture, I introduced you to the mutations, what are they and what are different types of mutation. And then in the last one, I taught you what is the molecular mechanism, how mutations happen in a cell. But now today I intend to teach you how and why mutations are induced by the chemicals and radiations. So I think we answer why first, and then we'll know how it is induced, right? So why would we introduce mutations in several organisms? So any guesses? Why would we like to introduce mutations in the several organisms? And before, I could have told you that, or we studied this earlier, that there are several mutagens. They occur in nature, and what we'll do for inducing mutation is we'll increase the mutation rate. Now, what is the mutation rate? We talked about that earlier. Mutation rate is the number of mutation coming in one generation. In spontaneous mutation, it is much less, but when we are trying to induce mutation, this rate is increased, and it becomes very likely that our desired mutation now will come and will be there in the next generation progeny. So back to my question, why would I like to introduce mutations? So one answer, we know this already, we discussed this in the previous class, was what? Is that we need to study mutations. First of all, we want an organism, and for modification we should know how to introduce them, because some changes we might like to bring in the progeny in the next generation. And otherwise also, for studying mutations, we need to know how to induce them and how to control them, meaning whatever number of mutations we want, only those should come. Say for example, we want point mutations, so we should know which mutant we will be using. Or maybe I want to randomly produce the duplications or major deletions. What should I use? I should know that. So it varies from purpose. What is my purpose? If my purpose is destroying someone, I would use a very strong mutagen which is going to destroy everything. So the purpose again comes in is that we want to study mutations, the changes, and we want to insert them wherever we want to. It may be for research purpose or it may be for application. Like we want certain changes to come in the next generation. Of course, we study mutations by crossing and waiting for next generation to come. But a very easy and a simple way could be but that's not simple. That's a very expensive and a way otherwise, hard, because we have a huge amount of DNA present in our cells. But another alternative method nowadays is DNA sequencing, that we know the sequences, and when we compare by multiple sequence alignments, then we can know where the change has occurred. But usually, since Mendel's times or classical genetics, we believe in progeny crosses and identifying where the mutations have occurred. So in this way, in this process, coming to mutations, we have learned two terms, though we discussed about them in the previous classes. One was mutagen. Mutagen is what? Is the chemical which we use for inducing mutations. And then we have mutation rate, which needs to be studied with for mutations. So I told you this already, but here again I tell you the name of few mutagens which we'll be talking about today in this class. One is base analog, for example 5-bromo-uracil. I'll be talking about the alkylating agent mustard gas. Then I will also talk about the intercalating agent, that is ethidium bromide. Then adduct forming agents like acetaldehyde or heterocyclic amines, and how these mutagens are linked to the cancer or maybe cancer treatment, that also we'll talk about. Then fifth mutagen which we'll be talking about is UV radiations, and I will also talk about the several other ionizing radiations which are used in inducing mutations. So let's begin with this. So first we talk about the base analogs. So what are base analogs? Base analogs are the halogen derivatives of the are basically the substitutes for purines and pyrimidines during nucleotide biosynthesis, and they behave differently. For example, and I said that they are usually halogen derivatives. So I cite one example, that is 5-bromo-uracil, and it is what? It's a derivative of uracil, but it behaves differently. Uracil normally binds to adenine, but if we add 5-bromo-uracil, it will behave as thymine. It will behave as thymine because it is halogenated at number 5 position of the pyrimidine. So when it behaves as thymine, what happens is that, or after the addition of bromo group to here, what happens is that it becomes very prone to tautomerism. So tautomerism we have learned very well in the last class with the help of several examples. So it becomes very prone to tautomerism, and what happens in that case is that when it is prone to tautomerism, it will change its keto form to enol form, and when it changes its keto form to enol form, what will happen? It will now bind to guanine instead of adenine. So with tautomerism, it will change its form from keto to enol, and the enol form binds to guanine. So what will happen after one round of replication? This will change to the AT base pair will change to GC transition result. So if we want to insert a transition mutation, that to a point mutation, these base analogs become a good agent. So we could use them, and they will give you point mutation and in after next generation. And another example of this is this base analog is 2-aminopurine. This also behaves the same way, and the 2-aminopurine, if it's there in the replicating solution, it will quickly go and occupy the position, and though it is purine, but it will pair with cytosine. Again, it leads to the change in AT pair to the GC pair. So basically these base analogs, they leads to transition mutations, and in the very next generation there is a change. And the good thing or the point to be noticed here is that they cause point mutations. So these point mutations we've learned already, they are quite important. They could lead to maybe substitution when it is a substitution only. So there we don't expect a change in the reading frame or the protein, but it may be a start codon again, or it may lead to the termination codon, and change in amino acid is obviously expected, right? So we go further, and next, first was we studied is what? Base analogs. How are base analogs important? So the next agent which I'm going to talk about is alkylating agents. So what are alkylating agents? Alkylating agents donate their alkyl group, as it's apparent from the name that they will donate their alkyl group to the amino or keto groups in the nucleosides. So in the nucleotides, so what will happen as a result of this? We have normal bases lying, but if their amino or keto group is changed and is maybe alkylated, the alkylating agents, this will do is that they will change again their base pairing affinities. And an example which alkylating agent you will use here is mustard gas, for example, sulfur containing mustard gas. And it was first chemical mutagen identified in the chemical warfare studies. So during World War, when the different chemicals or their effects were being discovered, it was observed that it's an important mutagen and it was used in the chemical warfare. So I said the alkylating agents will donate their alkyl group to the amino or keto group, and I gave you the one example, that is of mustard gas. And like for example, if it is guanine, at the number 6 position of guanine keto group, it will donate its alkyl group and it will change its binding affinity. So this 6-ethyl guanine, what will happen is it will base pair with cytosine. So you can see the binding affinities changes, and the alkylating agent which is popularly used for this purpose is ethyl methane sulfonate. So we use ethyl methane sulfonate for this purpose, and what it will do is it will change guanine to ethyl guanine, and this guanine now will bind to thymine. So we have what again, a GC base pair changed to a AG base pair. So these alkylating agents also are used for point mutation. So we got to classify, again I was saying in the beginning that it all depends on our purpose, what type of mutations we want. If we want smaller mutations, then we could use the point, meaning the very little small change in the organism. Again, that could also be very risky, because in the previous lectures we studied that they may be lethal also, they may be targeting some very important protein, but still if you want to insert a point mutation, we will use these alkylating agents. Then the next category of agents for inducing mutations is intercalating agents. So what are intercalating agents? The intercalating agents have the dimensions and shapes that allow them to wedge between the base pairs of DNA. That means what? Is that they will go and occupy the position between the two bases in a nucleotide, and then it will distort the DNA strand structure at that time, and it will change the functioning, maybe the transcription, replication, repair, all the functions of DNA would be changed because of this. So I said what? I said intercalating agents have dimension and shapes that allow them to wedge between base pairs of DNA. And an example of this intercalating agent is ethidium bromide. I hope you remember ethidium bromide we use where? You studied this with DNA studies, and you use it where? Any guesses? Anybody wants to guess? Where do we use ethidium bromide? Ethidium bromide, yes, you said it right. Ethidium bromide is a fluorescent compound that is commonly used in molecular biology laboratories during the purification and gel electrophoresis. The mutagenic properties of ethidium bromide, or maybe the UV light, whichever we use, it is used to, because it's a fluorescent agent, it is used commonly to visualize the DNA, the presence or the presence of DNA in the gels, meaning for identification of the DNA visualized by fluorescence. So ethidium bromide is one intercalating agent, and the intercalating agents, there are few other examples which I could use, but this intercalating agents, they are used for cancer chemotherapy also. One example which I like to give here is doxorubicin, and it is used to treat Hodgkin's lymphoma. And what happens here is that because the cancer cells undergo DNA replication more frequently than non-cancer cells, so they are more sensitive than the normal cells to these mutagens and damaging effects of these chemotherapeutic agents. So this property of intercalating agents make them very useful for DNA studies or maybe chemotherapy. But how they function, I told you, is that they have a dimension and shape which allows them to wedge between two nucleotides, where they alter the transcription, replication, repair, and other functions of the DNA, and this leads to DNA deletions, insertions, or maybe frame shift mutations. So it's not causing point mutation, so we reach to the higher, it may cause deletions, insertions, and lots of unpredictable things with frame shift mutations. Then another category of these mutagens which we are going to talk about today is adduct forming agents. So what are these adduct forming agents? The DNA adduct is a substance that covalently binds to DNA. It covalently binds to DNA and alters its conformation, and thus interferes with replication and repair of this DNA. So what is an adduct? It does what? It will covalently bind to DNA at wherever it wants to, and then it will interfere with everything. An example of this is acetaldehyde, and another one is HCAs, that is heterocyclic amines. So I said DNA adduct, it will interfere with everything happening in DNA. And the examples, I said acetaldehyde. I hope you remember acetaldehyde is a component in cigarette smoke, and that is how it causes cancer. So acetaldehyde is a component of cigarette smoke, and maybe that's a reason, but it interferes with the DNA replication and repair. HCA is another example which I gave you, is again a cancer causing chemical, and that are created during cooking of meats such as beef, chicken, and fish. So HCAs are formed at high temperatures from amino acids and creatinine, and they covalently bind to guanine bases, right? And at least 17 different HCAs have been linked to the development of cancer, such as those of stomach, colon, or breast. So these HCAs, they are produced if we cook, these beef, chicken, meats at very high temperature, and they are formed from amino acid and creatine. So, and how they function is that they form a adduct. They form a DNA adduct, and they will go and covalently bind to DNA, altering its conformation and interfering with the replication and repair of this DNA. So I hope these mutagens, by now you have understood that mutation is a very, very important phenomenon happening in the body, and the several of the cancer causing things, they overlap with these mutagens. So mutagens at several places, they are also causing cancer. So we come to the next mutagen, that is UV radiations. So it's a very common one, and everyone knows that the UV radiation causes the cancer. So I think a brief thing about UV radiation, all of you have seen, just I have brought this spectrum of light for you, and you can see that UV radiation is adjoining the visible range, and it has decreasing wavelength but increasing energy from the visible range. So and we already know that the UV light is absorbed by purine and pyrimidine bases at 260 nanometer. So these bases, they observe and get affected by UV light, and if we see the Earth's ozone layer, it absorbs the most dangerous type of UV radiations, and the UV radiations are sufficient enough to produce thousands of lesions in DNA per hour if any cell is exposed to this radiation. We know UV radiations are very, very harmful, but they are very important source of mutations, and it causes big mutations. The word which I used here is DNA lesions. They will produce DNA lesions inside the cell, and they are very dangerous, you know this. So the purine and pyrimidine bases absorb this UV light, and they have a lesser wavelength than visible light but more energy. So they have several effects, but if we talk about at the molecular level, they cause deletions, I told you already, and we use this for sterilization, so you know that it can cause the death of the organism also. And in the ozone, in the New Zealand, we know because of ozone hole. In nature, the clouds, they filter this UV light, but near the poles it is less filtered, and we have skin cancer happening because of UV light. And if we talk about the molecular level, we have formation of thymidine dimers. And what are these thymidine dimers? That the thymidine, when they are present together in the DNA, meaning at any place when we have two pyrimidines lying together, rather than bonding to the corresponding base, they form a bond between them. So as you can see in the picture is that we'll have dimers formed. So what will happen if a dimer is formed between two thymidine molecules? It will do what? It will not bind, and it will not be replicated from here. So it will distort the DNA conformation and inhibit the normal replication. So as a result, the gap will be there, and we'll have what? Errors being introduced during the replication. And if this, there is a relation between this dimerization, if the UV induced dimerization is extensive, it is said that at least it will lead to the killing of the organism. If depending upon the exposure, if this dimerization is extensive, it will lead to the killing of the organism. So UV light is very dangerous, so we will use UV light for mutation very, very carefully. More are the chances that it will have a killing effect. Rather, we use UV light for studying DNA, and we use it for sterilization more. So we now come to the last type of mutation which we were about to talk is ionizing radiation. Again, if we see them in the spectrum, they fall beyond the UV light, meaning they have wavelength still smaller than UV light, but they are more energetic than UV light. So we have three main things falling here is X-ray, gamma rays, cosmic rays. And cosmic rays, we talk about that a lot. So what they again fall in this category, and they are ionizing radiations. And what they do is that because they have higher energy, they are more energetic, they penetrate deeply into the tissues and causing the ionization of the molecules encountered along the way. As a result, the stable molecules and atoms in the cells are transformed into free radicals. The free radicals, we talked about that, the oxygen molecule, hyperactive in the last class, that they are quite an important source for causing mutations. So these high energy radiations, they are absorbed by the cells. They penetrate deeply into the tissues, and they produce what? They produce free radicals. And the free radicals, we talked about that earlier also, they can alter the purine and pyrimidine bases in DNA, breaking their phosphodiester bond, disrupting the integrity of chromosomes, producing a variety of chromosomal aberrations, deletions, translocations, and chromosomal fragmentation. So it will again produce big changes. So we could now categorize the mutants, like what is our purpose. If we want point mutations, we'll use the base analogs maybe. If we want a little more disruption, but if we want to kill, we use UV radiations. So with this one, probably that was the last one here. Yeah. So these are the 6 ways with which I told you how we can achieve mutations if we want to, and what mutagen we'll be using. It all depends on our purpose, what we want to do with in our study, or what is our requirement. So different mutagens have different properties. Right? So welcome again. You want me to summarize? I can summarize. We talked about 6 types of mutagens, and all of them have different properties, different regions. If you have any questions, you can ask me in the chat, in the comment box. I'll be very happy to answer your questions. As usual, like previous 2 classes, I'm back with another interesting question to present in front of you, and that makes that generates a curiosity among the students, is that whether they have understood it or not, or it will also tell me your presence that can you answer the question if it is indirectly asked in the examination. So do I read it out for you? It's there on the screen also, you can see. But the question which I am presenting to you is that we have a base analog, that is 2-aminopurine, right? So we are using what? 2-aminopurine, and it's a base analog for adenine. So during DNA replication, but it may base pair with cytosine. So we used a base analog for adenine, and it will pair with cytosine. And the base pair for bromouracil substitutes for thymidine, right? All right, right? But it may base pair with guanine, right? We have used a base analog for adenine that will pair with cytosine. We have used a base analog for thymidine that will pair with guanine. Follow the double-handed trinucleotide sequence shown here in the drawing, and follow that through 3 rounds of replication, assuming that in the first round both the analogs are present and they become incorporated wherever possible, right? So we assume that these analogs will be inserted in the first round of replication wherever it is possible for them. I have the sequence on the screen, maybe you could take it from there. So before the second and third round of replication, any unincorporated base analog is removed. So what does that mean? It means no incorporation has happened in the second and third round of replication. So can you tell me what is the final sequence that is going to occur in this mutation, where I have a sequence? I have shown you that on the screen, you can see it there. And I have added what? I have added aminopurine, which is substituting what? Which is substituting adenine. I have added 5-bromouracil, which is substituting thymidine. And the substituted adenine will pair with cytosine, and substituted bromouracil, it will, for thymidine, it will pair with guanine. So I look forward for your answer in the comment section. Maybe I hope few of you will give me the answers in the comment section, because this class, because of certain technical issues, I cannot bring in students here in between to answer me here. But still, it was a very lovely experience teaching you here in this mode. And with this one, I have finished off with mutations. I still can send you some important questions or maybe important segment where and in what form it is going to come in the exam. But in general, it becomes a very important segment of portion of molecular biology. So again, I will just wind up with this unit, and if you have any questions, you are most welcome to ask me. You have my email ID and you have my here itself you can ask me on YouTube comments section. Thank you very much for joining me here, and I love teaching you. Thank you very much. Love you.